Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us the 141st seminar, and I call it on University of British Columbia Day. Today's exciting day with Nobel Prize winner announced, the first Korean laureate in literature, and that is basically a few hours ago announced. Of course, this week is Nobel Prize week with exciting announcement regarding AI related recognition, including one of the synthetic biologists, Professor David Baker, and he got Nobel Prize in chemistry with some other AI people who enable prediction of protein or even de novo protein design. And also we are very excited, we see the continuation of RNA in regarding the cognition with the now microRNA. And last year, of course, we had the mRNA researcher with the COVID vaccine development, etc. And obviously, today's rising star speaker also under supervision or working with Jennifer Doudna. She obviously one of the pioneer who developed CRISPR system. So very exciting week. I'm very happy to host today's speaker today. And it's already October 10, and 17 days ago, I was in Munich to enjoy the October festival after attending a German conference, watching people drinking beer, listening to music, more importantly, enjoying the cool weather. Interestingly, last week in California, where I was attending the DOE JGI meeting, we saw 40 Celsius that week. Climate crisis is a reality, and we should do our best to solve the global problem by global collaboration. Fortunately, I started to lead an NSF Global Center called Circle as a center director, working with more than 40 academic investigators from 18 academic institutions from 6 nations, in addition to collaborating with 16 companies. In fact, I am now in Korea to discuss global collaboration, discussing the synergies between the global centers. I met and discussed multiple things with many pioneers and rising stars who gave SynBYSS seminars, including Dr. Jay Kisling, Nathan Hilson, Sang-Hyuk Lee, Humin Zhao, Byung-Kwan Jo, Song-Sun Im, Junyoung Park, Paul Freeman, and Yong-Soo Jin, all before gave a talk in this seminar series. I have been so excited to meet these people in Korea, as well as many leaders in the biofoundry. Okay, this is a little bit longer introduction today, because I was so excited because of the Nobel Prize week. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Professor Lindsay Eltis. He is a professor at the University of British Columbia and a Canadian research chair in microbial catabolism and biocatalysis, a very prestigious position in Canada. I have known him for many, many years. In 2022, he invited me to give a seminar at his university when I enjoyed great discussion and of course, one of the best food, including sushi, dim sum, seafood. Lindsay, thank you so much for your hospitality and great contribution to the scientific society, and the virtual podium is all yours now. And thank you again.
Thank you very much, Tae-Suk. So I'll just share my screen.
Sure.
Okay. Well, thanks a lot for that wonderful introduction. And thank you also for this opportunity to contribute to this symposium, which you've had going for several years now, and it's continued to flourish and bring people together and inspire the next generation. So as Tae-Suk said, I'm Lindsay Eltis. I'm in the Department of Microbiology and Immunology at UBC. I work on bacteria that transform or grow on aromatic compounds and steroids. And one of the applications that we're interested in is developing microbial cell factories to valorize lignin. The particular organism that we work on is a soil bacterium called Rhodococcus. It's closely related to mycobacteria and Corynebacteria. So it's got this outer membrane that contains mycolic acids. See if I can just get a pointer going here. Laser pointer, there we go. Yeah, so it's got this mycolic acid containing outer membrane. And the two things that are really kind of cool about this organism is one, it's got this tremendous, or that make it particularly cool for biocatalytic applications. That one, it's got tremendous catabolic capability, so it catabolizes an astounding array of organic compounds, but it also is actually used to produce thousands of tons per year of acrylamide, thanks to the activity of nitrile hydratase. So this is used in Japan and Germany to produce acrylamide. And the second thing that makes it kind of cool is that it's got this amazing biosynthetic capability as well. So this is an electron micrograph of Rhodococcus that's accumulated these lipid bodies. So if you starve it for nitrogen, it'll sequester its carbon, and up to 70% of its cellular dry weight can be triacylglyceride. So this can be harnessed. So we've got a couple of different objectives in our research. One of them is to harness the catabolic capabilities of this organism to efficiently convert lignin depolymerization products. So to expand them and tune them to particular feedstocks. And in addition, we develop tools to engineer rhodococci as biocatalysts. And this has included developing modular integrated vectors. And we've sort of used these to engineer the organism to synthesize wax esters, which is a high-value lipid. Why are we interested in lignin? It turns out that lignin is one of three components of biomass, it's a very underutilized component of biomass, and this is because it's this sort of heterogeneous aromatic polymer that's actually quite difficult to break down. So even though it's only 50 to 30%, it constitutes up to 30% of woody biomass. In most, so pulp and paper mills, for example, it's such as this one here that's just around the corner of where I do my research at UBC. It is burned. It's simply burned to drive provide energy for the pulping process. Canadian pulp mills typically produce about over 100 tons of lignin every single day, and so this is a really underutilized form of carbon. So the valorization of lignin really is a tremendous opportunity, a transformative and enabling one. I'm not going to go too much into the detail of what I do because I've only got a few minutes here, but basically the approaches that have a lot of, that are emerging that have tremendous promise for valorizing lignin involve these tandem processes, which involve chemical catalysis followed by biological catalysis. And so in the chemical catalytic step, the biomass is fractionated, and this produces a number of streams, including one for polysaccharides, but also a mixture of these lignin-derived compounds, so which are often monomeric, contain a high proportion of monomeric aromatic compounds. And these can be then in a second step, using a biological catalyst, can be converted to various value-added chemicals. So things like diacids, which can be used as building blocks to make polymers. So that's the basic idea behind what we do. Tai-Suk asked me to provide or give the members of this audience some advice or inspiration. So I think I'll just skip ahead to that. So and it turns out that I'm the graduate advisor, graduate director for the I'm the director of the graduate program in our department, and so I have to teach. I have a course that I give to graduate students in which I have to sort of tell them the types of things they should learn while they're in grad school. And preparing for this course, I came across this really nice short article that was written by Dr. Ramanujan Phage at MRC in England, and he sort of outlined these things that young researchers should learn, and I think they're very useful. And I won't go through them all because I'm sure you're very aware of some of them, such as you should learn how to write compelling prose and how to communicate properly. But I'd like to emphasize a couple of other things that I think are really worthwhile taking on board. Okay, so the first of these is to develop skills through mentored hands-on experience. And so this is really a lifelong process of learning from others. And I guess really a couple of things to keep in mind on here is, first of all, it's good to have a group of mentors because different people are good at different things. So you can have different mentors to meet your different needs, whether it's to help develop scientific strategy, technical skills, or you want mentoring on your career progression, or even your interpersonal relationships or leadership skills. So you should consider seeking mentors in each of these different areas. But when you're thinking about approaching someone as a mentor, you should also learn how to approach them. And in approaching them, or after you've decided you're going to approach somebody, you should prepare yourself for that meeting. So think through what it is you're hoping to get from them so that you can approach them the right way. And the other aspect to this mentoring thing aspect as well is to reciprocate and mentor others. So if you're a postdoc, for example, you should mentor graduate students and develop those skills. And if you're a graduate student, you can mentor undergraduates, and even undergrads in later years can mentor undergraduates in their first and second year. Okay, but this is really something that you can, if you've got like a group of people who mentor you, you can rely on them throughout your career, really. The other thing that's really important, I think, as a young researcher starting out, is to learn how to choose research problems. So this is understanding how to choose the research, or choosing the right sort of problem really depends on a combination of the timeliness of the problem, the importance of the problem, its feasibility, whether or not you have the resources to work on it, whether you're in the right scientific environment to work on it, whether there's competition to work on that, the plausibility, how well the project fits to your skill set, your capabilities, and your ambitions. But the other thing is that this is a dynamic process. So you can choose to work on, or what you work on throughout your career will change depending on what projects are, or how timely projects are, or how priorities change and how your own abilities change. I think that it's also really important to develop the creativity and the broader scientific vision to find problems worthy of pursuit, not just in your own field. And I think in this respect, as you, especially as you go through grad school and as a postdoc, you should understand how the different research projects were chosen that people are working on around you. And you can start by if you were given this project that you're working on in the lab you're in right now, you should understand how that particular project was chosen and developed if you didn't do that yourself. So that's a conversation worth having with your supervisor, the people around you. And finally, the last thing that I'd like to really encourage you to do is to learn how to conceive and design experiments. So that means not just crafting individual experiments, but also strategically stringing experiments together to work out the basis of complex phenomena. As part of that, when an experiment doesn't work, think about why it didn't work so that you can avoid repeating experiments, or why a particular experimental design wasn't optimal, and so how you can improve on it. I'm taking the time to highlight this because I think a lot of people invest time in learning particular techniques, and this is important, but it's also very ephemeral because techniques change. So actually, it's much more valuable to learn how to design experiments properly than it is to learn a particular technique. Okay, so that's all the advice, inspiration I was going to leave you with, and I imagine my time is up. So thank you very much for your attention and for this opportunity.
Thank you. It's absolutely brilliant. Thank you so much. I mean, there are multiple things resonate with me. I mean, you just mentioned you need to change your design approach. And actually, I also want to share, I used to use, so-called directed evolution to screen entire library. And at that time, I become the robot manually picking colony, up to 40,000 different colony, and then manually screened by multi-channel pipette. But now we could rely on, we should rely on AI, AlphaFold, whatever, that is also developed by Nobel Prize winner of this week. In that way, you can reduce the number of variant we need to screen. So when I finish my graduate work, too many colony I need to screen, I promised to myself I'm not gonna do protein engineering ever. But because of this AI tool, I actually started. And also I also use now liquid handler. In that way, even make things faster. So things change, and then that is one of the design we need to adopt, and then we need to learn about those kind of things. And the other thing I kind of realized that's a very important thing. You mentioned writing is important, and obviously that is true because that's the only way you could express or present your hardworking data And without kind of properly present, you cannot convince other people to, buy your story or claim in the paper. So absolutely, brilliant presentation you gave us. So thank you so much.
Thank you,
Okay.
Tae-Sik.
All right. So now the main speaker of today with a longer introduction. Dr. Brady Cress is a group leader at Innovative Genomics Institute in University of California, Berkeley. His lab specializes in the development of microbiome editing technologies. As part of the Innovative Genomics Institute's Berkeley Institute for Optimized Microbiome Editing, in short, BIOME, research in his lab aims to engineer microbial communities to address global challenges associated with human health and climate change. Prior to his current role, Brady completed an NIH F32 postdoctoral fellowship in Jennifer Donna's lab at UC Berkeley, where he developed CRISPR-associated transposases for species and site-specific genome editing within microbial community. This work is one of my favorite reports published at the similar time as my lab's PNAS paper covering a similar microbiome engineering tool, drawing on a strong foundation in genome editing and forward-thinking approaches to microbiome research. He and his lab are at the front of harnessing the power of microbial communities to shape a healthier and more sustainable future. Interestingly, I met him when he was a graduate student in the Matteo Hoffa's lab at RPI, and his CRISPR paper with Matteo is also one of my favorite papers. Last year, he kindly invited me to give a seminar at UC Berkeley, and then show me around the IGI building, including Jennifer Donna's office and her awards. Amazing, a lot of awards, except for the her Nobel Prize, unfortunately, because she was not in her office at the time. Brady is truly a rising star and future leader, and I am so excited to listen to his talk today, and that's why I'm introducing him too long, I guess. Brady, thanks so much for your time today, and please take it away and keep me awake until 1 a.m. and later today with your exciting talk. Thank you.
Thank you very much, Tae-Sik. That was a very, very kind introduction. I'm just going to set a timer here to make sure that I'm...
No, you don't need to worry about, I never caught anybody because I'm so excited always listen to your talk. So never caught, so don't worry.
All right, well, I'm going to go ahead and share screen. Okay, can you all see that?
Yes, go ahead.
Perfect. So I didn't check to see who all's on Zoom in case we have a crowd. Hello, everybody. I think probably different time zones. So good morning, at least from Berkeley, where I'm located. Thanks to Tae-Sik for the invitation. And I also just want to give him a shout out for what I think is tireless efforts in initiating and maintaining the seminar series for several years now. And for the links that you go to showcase young scientists. Also, quick shout out, Lindsay. Thank you very much for joining, highlighting your lab's exciting research, as well as the really nice tips on seeking mentors, which I fully agree with being incredibly important in science, and picking the right scientific problems. So I would really encourage people to think deeply about that. So today, I'd like to tell you two stories. First, I'm going to tell you about our efforts during my postdoc in Jennifer Doudna's lab to develop what we kind of think of as a generalizable tool set for precision microbiome editing. And then I'm going to transition into telling you about our goal to unlock the power of phages, or the viruses of bacteria, as genome editing vectors for microbiome engineering. But before I begin, I just want to give a very quick introduction to my lab. So we are situated, as Tae-Suk mentioned, in the Innovative Genomics Institute at the University of California, Berkeley. And my lab is a founding lab of Biome, or the Berkeley Initiative for Optimized Microbiome Editing, which is spearheaded by my postdoc mentor, Jennifer Doudna, as well as Jill Banfield. And under the Biome umbrella, my lab is largely focused on the development of microbiome editing technologies that we believe are required to affect meaningful changes in microbiomes to tackle very big global health and environmental challenges. I'd also like to give a quick shout out to some young PI colleagues here at Biome that run basically neighboring labs in the building. The first is Ben Rubin, who was a postdoc lab mate in Jennifer's lab and a really close colleague that co-led some of the work that I'll be describing shortly. Spencer Diamond, who also co-led this work with Ben and I, while he was a postdoc in Jill Banfield's lab. And so we kind of like to think of this work as a merging of two big fields, precision genome editing with genome-resolved metagenomics, and combining these two into the new field of microbiome editing. Also, Carlotta Ronda, who's a very talented new PI in Biome that just published similar work to what I'll describe first today during her postdoc in Harris Wong's lab at Columbia. And then finally, another colleague of ours, Rohan Sachdeva, who was a postdoc in Jill Banfield's lab and is the director of the computational infrastructure here, and also just a brilliant scientist and close collaborator on a lot of fronts. So without further ado, I'd like to begin really by motivating our lab's interest and Biome's interest in microbiomes. Experimental genetics on microorganisms, which we think of as the ability to manipulate genes and study the effect of those manipulations, has been very powerful as a tool for understanding microbes over the last half century. But it has been almost exclusively focused on isolates, despite every microbe that matters for us really living in the context of a microbial community. In fact, microbiomes and not the microbes in isolation are critical for functioning ecosystems, and they're key actors in human, animal, and plant physiology. For example, many people are familiar with the statistic that the human body contains on the order of 1 bacterial cell for every human cell. And the community structure of these microbes plays really important roles in things like development. It can contribute to many human diseases, also impacts how our bodies respond to certain medications. In many cases, these communities exert really strong influences on things like the climate through methane production, in this example of a ruminant animal. Similarly, this happens in rice paddies, where microbial communities that are associated with the plant roots also provide benefits to the plant through access to key nutrients. Microbiomes are also instrumental in producing some of our favorite foods. And I could go on and on with examples, but I hope we can all agree that all of these communities share a common factor, and that is that their functions derive from the interactions between community members. So I think if we hope to exert precise control over microbiomes to address health challenges and climate change, we need tools to achieve precise DNA editing within microbiomes. Furthermore, establishing experimental genetics in microbial communities should also help to close some major knowledge gaps in biology. This is a phylogenetic tree of life that was constructed by Jill Banfield's lab, where each of these red dots on the phylogenetic tree represents a phylum with no isolated representatives, and you can see red dots everywhere. This suggests that if isolation of microbes continues to be a prerequisite for genetics, then we're not really going to be able to do experimental genetics on the vast majority of the tree of life. And potentially most important, performing genetics within microbiomes, even when we can't isolate its members, but we can cultivate the community in lab, should allow us to study genes involved in inter-organism interactions. So to close these knowledge gaps in the uncultivated microbial majority, And ultimately, to control natural microbial communities, we need a tool set for effective microbiome editing. And so with this in mind, my postdoc colleagues and I set out to create a generalizable tool set to enable species and site-specific DNA editing within microbial communities. And the first step in this process is just determining which organisms in a community are genetically accessible. So in this approach that we called environmental transformation sequencing, or I'll often refer to it as ET-Seq, this was largely developed and implemented by Ben Rubin, my postdoc colleague. This involves a non-targeted edit that's delivered into a community by an untargeted transposase. And then sequencing is used to kind of inspect where that genome edit went in to give us an idea of which organisms in that microbial community were genetically accessible. We can then leverage that information of what can be edited within a community to achieve species and site-specific editing using a targeted transposase within microbial communities. And I'll first explain how we determine which microbes in a community are editable using ET-Seq. So in ET-Seq, a non-targeted transposon is simply delivered into the community, and it randomly integrates a trackable piece of DNA. We often refer to this as a barcoded DNA. And without any outgrowth or selection, we can simply harvest the DNA and sequence the junctions between the transposon and the host DNA. This tells us which organisms received insertions. And then the amount of insertions is measured in each host, and it gives us an understanding or a quantification of how genetically accessible each organism is. This is then ultimately normalized by the relative abundance of each organism so that insertion efficiency doesn't look really good in something just because it's really abundant. And so really the conclusion of this pipeline is that it tells us, it gives us a quantitative measure of genetic accessibility of, in theory, all members of a microbial community, as long as they're at a reasonable abundance. And I just want to point out before we move on that, Ben spearheaded the development of ET-Seq and put a huge amount of effort into optimizing this protocol, which ultimately was really amplifying needles out of a metagenomic haystack for many of the species in this community. So a really important and tough problem. And then our colleague Spencer Diamond, who I mentioned was a postdoc in Jill Banfield's lab at the time, developed a huge amount of really powerful custom code to analyze and quantify all of this sequencing data for this work. So we really set out first to validate ET-Seq with an analyte spike-in experiment. And to do this, we prepared mixtures containing known fractions of a pre-edited Klebsiella strain with its unedited wild-type counterpart. And we mixed this in ratios spanning several orders of magnitude. These mixtures were then spiked into a synthetic 9-member soil community, which I'll discuss again later. That created effectively a standard curve of edited Klebsiella within this more complex community. And then we assayed these DNA edits using ET-Seq. So this plot on the right shows the ET-Seq derived fraction of cells that contain measurable insertions on the y-axis, which is very closely correlated with the known fraction of edited Klebsiella that we spiked into each sample. This experiment also indicated to us that we could pretty reproducibly detect one edited cell out of 1 million total community cells. And this is all without relying on selection to enrich, which is sort of a prerequisite for how we think about measuring edits these days. So now that we had a tool to quantify the fraction of cells with genomic insertions inside of a mixture of cells, we set out to actually test editing within a community. So for the first test of ET-Seq within a community, we assayed the genetic accessibility of this 9-member synthetic soil community that I just mentioned by multiple delivery methods. So we tested delivery by conjugation, which involves transfer of a plasmid from a donor E. coli strain into the community. We tested electroporation as well as natural transformation, where we just add the DNA to the community and see if any of the members take up the DNA. And our expectation was that we'd be able to measure or at least determine which delivery method was feasible for each of the organisms in this community. Okay, so in this hypothetical plot, this basically shows that for the red organism, conjugation worked really well. Worked really poorly, actually, while electroporation worked really well. Diving into the actual data from this experiment in the community, we were able to we were able to measure delivery consistently for all of the members that constituted at least 1% relative abundance. So that shows that one limitation of this method is just organisms that are very low abundance either suffer from delivery incapability or we just can't measure it. We also noted some differences between the efficacy of each of the different delivery approaches. And so, for example, Burkholderia caledonica was able to be edited through delivery by electroporation, but not by conjugation or natural transformation. And so at this point, we were pretty happy to find that this approach could achieve what we really needed it to, which was simply to reproducibly measure genetic accessibility of reasonably abundant community members. So coming back to this general framework for microbiome editing, we felt pretty happy that we had developed and demonstrated ET-Seq as a way to characterize what was genetically tractable in a community. But I want to transition now to talking about how we've developed targeted transposases to implement editing in community members that we previously measured through ET-Seq to be tractable. Okay, so to take advantage of ET-Seq as a roadmap for performing targeted genome editing in a microbiome, we needed an effective DNA editing tool. And so at this point, we turned to a really fascinating natural mobile genetic element known as a CRISPR-Cas transposase. Around the time we started this work, there were two papers that were quite seminal that described two different variants of these systems experimentally, although they had been known to exist bioinformatically for several years. And so this work really enabled us to perform site-specific editing in communities. So for the people that in the audience that may not be familiar with these systems, they are TN7-like transposases that have effectively co-opted nuclease-deficient CRISPR-Cas proteins for RNA-guided DNA integration. So you can think of CRISPR-Cas systems that don't cut DNA, but just guide to a particular site in the genome, which then recruits the transposase to insert genetic cargo or a large piece of DNA at that site. For that reason, we thought these systems would be really useful for performing targeted editing in specific species, which we could use for both breaking genes by disrupting them or integrating large pieces of DNA. And one of the reasons we thought this was particularly useful is because unlike other common CRISPR tools like Cas9 and Cas12, performing genome editing with Cas transposases doesn't rely on inefficient DNA repair mechanisms in bacteria like homologous recombination or non-homologous end joining. So whereas Cas9 and 12 have evolved to cut and kill bacterial cells or mobile genetic elements, Cas transposases have really evolved to survive that event rather than cutting DNA. So with this in mind, we set out to build what we refer to as DARTs, or DNA editing all-in-one RNA-guided transposase systems. As shown by environmental transformation sequencing, we knew that electroporation and conjugation were broadly effective and should be ideal delivery strategies for the 9-member synthetic community and probably many microbial communities. So we constructed mobilizable suicide vectors that have everything needed for conjugation and programmable genome integration, all on a single plasmid. And then we benchmarked these all-in-one systems by integrating an antibiotic cassette into a particular locus of E. coli delivered by conjugation. And the data that I'm highlighting here on this slide corresponds to the type 1F CRISPR TN7 system from Vibrio cholera, which we refer to as VC-DART, because this system really turned out to be the most accurate in our hands by far. Doesn't mean the other system's not useful for certain things. This one was the most precise. And after selective outgrowth, we performed environmental transformation sequencing to quantify target site specificity. This plot is basically showing that 100% of the detected insertions across E. coli's genome out of thousands of unique insertions were precisely on target at the site programmed by the guide RNA. So this indicated that we had the beginning of a really powerful tool for targeted editing in microbial communities. On our mission to find the ultimate programmable and precise microbiome editing tool, we tested a couple of other systems, including another RNA-guided CRISPR-associated transposase. And this really just shows the benchmarking between VC-DART and another prototypical Cas transposase called SH-DART. What you can see in the plot here in the bottom left is SH-DART had a very low fraction of on-target insertions. So a lot of insertions, but not many of them were on target. And you can really see this exemplified in this genome-wide plot showing a bunch of low-frequency edits across the genome with SH-DART with an enrichment at the target site. But for microbiome editing, we felt this was really not very practical. So with this in mind, we moved forward with the VC-DART system rather than SH-DART. Next, we set out to test targeted editing in this synthetic 9-member community. And we expected that selections in most natural communities would be quite challenging, just because traditional mode of selection is through antibiotic marker insertion, followed by antibiotic selection. But antibiotic resistances are quite prevalent in natural microbial community, as well as they tend to emerge spontaneously, or mutants will evolve spontaneous resistance. So we wanted to know first if ET-Seq could detect these DART-mediated edits in the absence of any form of selection, just performing the edit and applying ET-Seq to see if we had on-target editing. So in this experiment, we basically used DART rather to deliver a barcoded transposon, so no selective marker, just a piece of DNA containing a readout that we could read by PCR. And we conjugated this into the community using 2 different guide RNAs on the Klebsiella genome. And this plot is just highlighting that we could actually detect both of the on-target insertions across the Klebsiella genome exactly where we expected them. This top inset is just highlighting with asterisks where we had actually targeted the guide RNAs. And you can see that in absence of a selection like we had done on the previous slide, we do have more noise. And I don't think these are real off-target insertions. We can get into details later if you're interested. But inability to filter out bioinformatic artifacts from sequencing, we see some low level of noise of insertions across a relatively abundant species in this community. So we thought this was a super encouraging result because really what we're observing here for the first time are targeted genome edits made within a non-model organism inside of a community and all without selection. And so to further show that targeted edits could be applied in a meaningful way in a community context, we wanted to make selectable edits in some of the more abundant members, Klebsiella and Pseudomonas, to change the composition of the community using selection, biasing towards the edited Klebsiella or biasing towards the edited Pseudomonas. And the experimental setup is just shown here on the left, where we introduce DART by conjugation, as we've done before. But in this case, we're delivering an antibiotic resistance gene to either Klebsiella or to Pseudomonas. And as I kind of alluded to earlier, we found that even in this really simple, small community, it could collectively resist most of the different antibiotics and antibiotic combinations we threw at it. And so what this meant is we needed to also capitalize not just on the insertion of an antibiotic marker, but also on counter selection mediated by specifically breaking a counter-selectable gene called PIR-F in the genome of these organisms. And so the plot on the right basically just demonstrates that this approach was quite successful. So the left 3 replicates are the community before delivery of the DNA. And in the middle series, we show triplicates of Klebsiella in a guide RNA targeting Klebsiella, followed by enrichment on the antibiotic and counter-selective agent. And we basically convert the entire community into Klebsiella by enrichment of only Klebsiella relative to the rest of the members. And the set on the far right shows something very similar, in that if we just switch this 30 base pair piece of DNA that programs the system to go to a different locus in a different organism, we can enrich for the only the targeted organism and none of the other bacteria in the community. So the take-home message here is really that compared to previous state-of-the-art integration methods that rely on long homology arms, we could dramatically reshape the microbial community in different ways, in this case, just by changing a 30 base pair guide RNA sequence. Okay, so we next tested DART as a means to confer a new metabolic capability to a targeted organism in a microbiome. When DART cargo contains antibiotic markers, it enables positive selection of edited cells by application of that antibiotic, but it negatively selects against any unedited community members that lack that resistance. Conversely, if we apply DART with a cargo containing a nutrient utilization pathway, we should be able to provide a metabolic niche for edited cells while minimally impacting untargeted members. And so to demonstrate targeted metabolic pathway integration, we used Cas transposases to provide, in this case, Pseudomonas simiae with lactose assimilation capacity inside of a 4-member synthetic community that was otherwise unable to metabolize lactose. And as you can see in this plot just showing the composition of the community before and after enrichment on lactose, we were able to largely enrich for Pseudomonas simiae, basically facilitating its almost complete enrichment over the rest of the community, with the exception of Ralstonia, which we think was likely able to persist slightly because of cross-feeding, although it's possible that there was an off-target edit in Ralstonia. Ultimately, we envision that these types of nutrient utilization or biosynthetic marker cassettes will be very important for enriching and engrafting Cas transposase-delivered genetic cargo in the context of these complex microbial communities. Okay, in the interest of time, I'm just going to highlight on this slide that we wanted to move beyond synthetic communities and move towards natural communities. And so what we've done is taken an enrichment from an infant stool sample. So this is a real microbial community derived from a real human sample. And this was provided by the Banfield lab. And we simply wanted to demonstrate that in the context of a real community, we could apply genome-resolved metagenomics to identify organisms in this community without ever needing to isolate, design guide RNAs, and target unique members in this microbial community for site-specific editing, followed by enrichment of those members without ever having to pre-isolate or pre-sequence individual isolates from that community. And so what we're showing here are 2 genetic loci from 2 of the E. coli strains in this community. After performing site-specific editing with VC DART in both of these strains, followed by an antibiotic selection scheme, you can see that we do a really great job of targeting one of these organisms and selectively enriching for it, and a pretty good job, although not perfect, for the second lower abundance E. coli strain that we targeted. So in this way, I hope I've convinced you that we can enrich through traits of interest or differential loci in different strains that are otherwise quite similar within natural microbial communities. Okay, I'm going to skip this slide and move on to the conclusions of this section. The key points that I hope you'll take away from this work is, first, we've developed environmental transformation sequencing for detection of genome edits in microbial communities without relying on selection. Next, we've used ET-Seq to identify successful delivery strategies and genetically tractable community members. We've also developed DART systems for species and locus-specific genome editing in microbial communities, including in non-model strains. And we've shown that DART can be used to manipulate the structure of microbiomes in simple and programmable fashions. So altogether, we think this does represent a very generalizable approach for community editing. However, despite all that we learned in this process and all that we were excited to accomplish, one of the most important lessons was that delivering DNA into diverse bacteria is incredibly challenging. Fortunately, nature's provided us with a vast and largely untapped reservoir of natural DNA delivery systems that we could, in theory, harness to deliver our editing tools. And so my lab aims to harvest and repurpose natural mobile genetic elements like phages and plasmids to increase the taxonomic breadth of organisms that we can deliver to, and ultimately, in turn, improve our microbiome editing efficiencies to the levels that are needed for real-world applications. One One of the classes of delivery vectors that we are very excited about harnessing is bacteriophages, which have evolved to be very effective DNA delivery machines. Phages are the most abundant biological entities on the planet. And despite that, the function of the vast majority of their genetic content remains a mystery. And until we can unlock the secrets of their genomes and understand which parts of their genomes are important or which parts we can get rid of, engineering and harnessing them and their genetic components for microbiome editing will remain a huge challenge. So this brings us to a hard left turn in my talk today, which is focusing on phage functional genomics, or experimental genetics approaches to help us answer the question, what is important for phage infection? Before I dive into the details, I just want to recognize the collaborators that really made this work possible. A couple years ago, Ben Adler and I, who is a postdoc in the Doudna lab, and at the time we were postdocs together, we set out to develop a series of phage functional genomics technologies. And I'm just highlighting one of those approaches in today's talk. But I think we've made a lot of great progress and continue to collaborate on this now that I'm running my own lab. The work that I'll discuss today also involves Munthar Al-Shamari, a PhD student in the Doudna lab, who's also a former lab mate of mine. And this was co-led by all of us. Okay, so at the outset of this work, we really set out three major objectives to establish an effective phage functional genomics pipeline. First, we wanted the approach to be effective across phage diversity, and phages are very diverse. We also wanted the approach to be scalable across the genome. And finally, we wanted a highly multiplexed, cheap, fast readout. And so for all of these reasons, we settled on a transcriptome-wide pooled CRISPR interference approach that satisfies all of these constraints. Thanks to some really incredible work by Ben Adler earlier in his postdoc, which is cited below if you'd like to read it, we suspected that the key to effective delivery, efficacy of a phage functional genomics tool against diverse phages would be in targeting the RNA of these phages. Given that RNA is a conserved, essential, and likely for phages vulnerable, as shown in this schematic here, we basically set out to develop a transcriptome scale knockdown approach that could target every gene in a phage genome through the accessible mRNA, or in the case of single-stranded RNA phages, through its genome itself. In theory, this should allow targeting of RNA phages, nucleus-forming jumbo phages that otherwise protect their DNA from CRISPR enzymes, phages that have heavily covalently and epigenetically modified genomes that prevent efficient binding by DNA targeting CRISPR tools, and otherwise single-stranded DNA and double-stranded DNA phages that express their genes through RNA. Thanks to work by a former PhD colleague, Emmerich Charles, in the Doudna lab, we knew that the RNA-guided RNA binding protein, Cas13d, was very well suited for targeted translational knockdown in E. coli, where we anticipate that this works by binding the ribosome binding site and blocking translation initiation. So by doing this, you can prevent expression of the targeted gene. But to test if the ribosome binding site was uniquely susceptible to targeting by Cas13, we tiled guide RNAs at 1 nucleotide resolution across the transcripts of several essential E. coli genes, and ultimately observed that there's a strong Cas13d dependent fitness defect when guides are targeted primarily at the translational start site. And so this confirmed our hypothesis that by occluding access of the ribosome to the translational start site, that we should be able to inhibit translation in a programmable fashion. This plot on the bottom right basically just highlights a design rule for us, which is if you'd like to target a gene for translational repression, you should target within plus or minus 20 base pairs of the translational start site. Okay, so Ben took this knowledge and applied it first against model phage T4, which encodes a glucosylated genome that we expected to be problematic for DNA targeting tools. So in these phage plaque assays, successfully knocking down essential genes, but not non-essential genes, should reduce plaquing. So if we can shut off, for example, expression of the major capsid protein of a phage, it shouldn't be able to form a plaque. And this is exactly what Ben observed with dCAS13. When targeting essential genes in red, we could reduce infection, but targeting non-essential genes in blue did not reduce infection. In contrast, DNA targeting tools like dCAS12a or dCAS9 were largely ineffective. Next, we scaled this across many diverse phages. There's a lot of detail in this network graph, but the key takeaway is that Ben has a really great collection of diverse phages spanning multiple Baltimore classes, double-stranded RNA, single-stranded DNA, and single-stranded RNA, chemically modified genomes, temperate, chronic, and virulent lifestyles, including a nucleus-forming jumbo phage called Gosler. So in this experiment, Ben tested 2 guide RNAs targeting the essential major capsid protein for each phage, where darker blue indicates very strong fold reduction in phage plaque count, meaning reduced infection. And for some phages like T5, you'll note a marginal reduction in plaque counts. But for these cases, at least one of those guide RNAs leads to large reductions in plaque area, which also suggests a reduction in phage infectivity. So for each of these cases, we can see by targeting this essential gene major capsid protein across these diverse phages that this tool is largely effective. And I just want to give a really quick shout out to Emily Armbruster, who's a good friend and collaborator of ours from the Poliano lab at UCSD, who actually ran these experiments with this jumbo-forming nucleus phage Gosler. Okay, so now that we knew that we had a tool that was effective across phage diversity, we expanded this technology to the genome scale, basically targeting every coding sequence in the phage transcriptome. So in this approach, we design guide RNA libraries with 7 guides targeting each coding sequence at the ribosome binding site region. Then we induce expression of these CRISPRI-ART libraries in E. coli, adsorb phage, and then after the phage has adsorbed to these cells but not infected and started an infection cycle, we plate them quickly onto selective media with inducer. And this basically allows micro-competition experiments where each cell that lands as an individual colony will start competing with the phage that's adsorbed onto it. And in the morning, after overnight incubation, we can scrape the colonies up into a pool and sequence the guide RNAs. In this case, we expect guide RNAs that were effective at suppressing phage infection by targeting essential phage genes. These should form into colonies because E. coli can survive. Guide RNAs that target non-essential phage genes don't protect E. coli, and therefore those colonies shouldn't form, and those guides should be depleted. And so we can compare to a control where no phage was added, basically our expectation for a naive library. And this allows us to infer which phage genes are critical for phage infection. This is what the data looks like for model phage T4, which is the most well-studied virulent phage. And it's a lot of data, so I'll just draw your attention to the upper right quadrant of this plot where we're highlighting phage T4 genes that were measured to be important for infection and which are likely essential for T4. Thanks to decades of work on generating single T4 mutants, we also have a really nice ground truth of gene essentiality for comparison for many of the genes in T4's genome, although many of them still have unknown essentiality. And you can see through this plot that we do a pretty good job of distinguishing known essential and non-essential genes, although it's not quite perfect. But I'd like to highlight that in contrast to decades of experiments, this data set was determined all in a single and really quite fast experiment. And so using this information, we can generate these genome scale maps with the genes represented on the outside and their corresponding fitnesses on the ring on the inside. You can tell with this visualization, for example, that regions that are enriched in structural genes in dark blue that form the virion that's required for packaging of the genome and reinfecting a new cell are very, very clearly essential, which is known for T4 and many other phages. Finally, we can apply this approach to diverse phages. And really, I'm just highlighting kind of a spectrum of modelness. On the far left, we have T5, which is another model phage, although not as well known as T4. or as well characterized as T4. In the middle, we have a non-model phage called SUSP1. And on the far right, we have a very non-model phage that we refer to as a singleton because most of its genes have no known homology to any other gene that exists in public databases. And despite that, you can see that we still do a pretty great job of being able to call out which regions of this phage genome are important, which genes are likely essential. And a couple of takeaways from this is, from an applied perspective, in all of these phages, there seems to be at least one long tract of non-essential genes that we might be able to delete and add custom microbiome editing payloads into the phages for customized delivery. From a basic biology perspective, there's some interesting insight that we've gleaned. For example, in this very non-model singleton phage, PTXU04, there's a single huge gene of 7,000 base pairs that takes up more than 10% of this phage's genome, and it's absolutely essential for this phage. And yet, we have no idea what its function is because it is not annotatable. So, in conclusion, CRISPRI-ART enables simple measurement of phage gene fitness at genome scale, and our hope is that others will use it to shine spotlights on interesting genes that are important for phage life cycles. And with that, I'd like to conclude and thank everybody for your attention. And again, thanking Tae-Suk and Lindsay. Huge shout out to everybody in my lab. We're a small but quickly growing lab. And all of those people in my lab have contributed in some form or fashion, with those bolded actually playing an experimental role in this paper. Also, thanks to my colleagues in the IGI who are running small but growing labs in a similar fashion to mine. And especially my colleagues in the Doudna lab, Ben Adler and Munthar Al-Shamari, who co-led this work, who are just phenomenal scientists. And I will note that Ben Adler is going to be a steal for whichever department snaps him up as faculty. So keep an eye on him and check out his research if you haven't yet. Also to our collaborators at UCSD on some of the phage work at the Poliano lab. Also, massive shout out to our funding sources for both past and present for the work that I've shown here. And with that, I'd be happy to take any questions. Okay, so should I check the chat potentially?
Yeah, Brady, I saw there's a question from somebody in the chat.
Great.
Actually, it's in Q&A. I can read it to you if you like.
Got it. Yeah,
Okay.
so the question is,
Yeah,
just
go ahead.
For those that can't see the question is from Jonathan Suganda. And for ET-Seq, are there multiple transposases? Are the readouts limited by the specific sites the barcodes integrate into? For including nutrient utilization pathways, does editing efficiency decrease as cargo length increases? These are all really great questions. Let me start with the first. For ET-Seq, there's no limitation on the type of transposase that can be used. We typically use a Mariner derivative, HIMAR, which is reasonably untargeted or non-targeted, and TN5, which is also reasonably non-targeted. And none of these are perfectly untargeted. There is some slight bias, but yeah, I think generally there's probably not a huge limitation in the types of spots that these two random transposases can jump. There's probably some slight bias, but in terms of just understanding if a single edit can happen anywhere in the genome, I doubt that it's limited on a species by species basis. Are the readouts limited by the specific sites the barcodes integrate into? Maybe if you could clarify slightly, my assumption is what this question is asking is, are there sites that are where barcodes integrate that are maybe recalcitrant to the library prep or to PCRing the insertion out of the genome. I think that's feasible. We know there are sites in genomes that are very challenging to amplify. And so that is potentially one limitation. We didn't really inspect this too much from an experimental perspective. The third question, for including nutrient utilization pathways, does editing efficiency decrease as cargo length increases? We have tried up to 10 kilobase pairs with these particular targeted transposases, at least through the mechanism that we're delivering currently, which is a suicide vector that cannot replicate when it's received inside the recipient cell. Through conjugative delivery, we don't see any substantial decrease in cargo efficiency with increasing cargo size. We've not really tried anything extremely large, something we'd love to do. And I'm not actually, I should know this, but I don't know what the largest tested in literature is. What I will say is these natural transposons are ranging in size up to, let's say, approximately 100,000 base pairs. So our expectation is that they can mobilize just fine. We just don't know if there's a significant loss in efficiency. Hope I answered those and understood the second one in particular, but please follow up if I misunderstood.
Okay, so I'm sorry, you know, I have some technical difficulty after you finish your talk, so I cannot jumping in. So, by the way, brilliant talk. I actually want. So, Lindsay, do you have any, you know, more question, more comment?
Me?
Yeah.
Yeah. No, no, I thought that was a terrific talk. Yeah, I have a few questions, but I don't know if it's the first time I've heard the term unannotatable. What did you mean by that?
Yeah, it's probably an imprecise term. I should be careful. No domains of known function, only one known domain in the entire 7 kilobase pair protein. Maybe I referred to unannotatable in many places, but basically known domains of known function.
I was also curious about some of those when you were doing that experiment to targeting the capsid protein. Why do you think targeting the capsid protein in some of those phages didn't affect their viability very much, like the T5?
Yeah, so I think there's several reasons. One is for T5 in particular, T5 injects its genome in 2 steps. There's a handful of genes, around 10 kilobase pairs, that is injected first. And it's believed that that does a very good job of basically chopping up the entire E. coli genome, probably plasmid DNA that's there as well, with an exonuclease. And so we think that probably some phages that have these kind of specialized approaches to protect the majority of their genome, including the major capsid protein, are probably doing a good job of suppressing the tool enough that we can just barely detect a reasonable signal. There's probably other mechanisms that phages are using to similarly make it hard to knock down infection by targeting the major capsid protein. One in particular is potentially, although we haven't explored this deeply, genes that are expressed in a particular stage of a phage's transcriptional program, whether it be early, middle, or late, might be more or less susceptible to targeting. Yeah, I'll think about it and get back to you if I have any other thoughts here. But T5, we think we have a pretty good explanation for, at least.
Yeah, I'm not a bacteriophage person, so it was really interesting to see that, but I had no really insight into like why that might be. So cool. What percentage of a microbiome do you think you're able to target with the initial 3 approaches that you were using? So before you got into the bacteriophages, the electroporation and conjugation, and do you have a feel for that?
Broadly, I mean, so I guess maybe one caveat is in those experiments, we tested a handful of communities that were not very
Yeah.
complex.
Yeah.
I think you're probably more interested maybe in like broadly across the microbial tree of life, what fraction do we think we can currently hit? Maybe I would make one caveat to that question first, which is if we'd like to do effective microbiome editing, we need very efficient delivery and editing. And so if we can get one colony on a plate, is that really effective for the things that we need to do? And should we call that microbiome editing? So I think probably, I don't have a good percentage for you. I think across the entire microbial tree of life, including many uncultivated candidate phyla radiation, we probably are hitting a very minute percentage. And what I will say is that is with the current delivery technology. So an RP4 conjugative system. There are many conjugative systems that we could in theory harness, many phages that we know can infect bacteria because we see them integrated as prophages. And so I think ultimately, if we can attach our DNA editors to those, we'll have a much better shot at accessing the entire microbial tree of life. For now, with the limited set of tools that we started with, I think we are very limited in the diversity of taxa that we can transform into.
Great. Thanks.
Okay, so even though we a little bit, the seminar become longer, I want to ask one question. This is very important question to me for my research and for your research as well. So I'm not sure you also envision the same way I envision the future microbiome engineering. So what I wanted to do is something like this. So right now you use the synthetic community to demonstrate your tool's utility. And then I believe you use the phage. That's a brilliant idea because that basically kind of overcome the limitation of those kind of DNA delivery. And that is also the one limitation, my method using the liposome, I guess. The question I wanted to ask is, one day I want to do real practical genome the microbiome engineering, without any limitations, something like this. This is my kind of protocol. So we basically have, let's say, billion different bacteria in the community, and then we do sequencing all of that, metagenome sequencing, whatever future next generation sequencing, know most of the sequence. And then let's say we found some interesting bug we wanted to target, and then engineer or even kill, whatever. And then design the guide RNA that target those things unique way, and then do the whatever engineering. One caveat, one limitation might be the bacteria may not entertain the Cas9 or CRISPR system, whatever, transposase and blah, blah, blah. And that's one challenge is. And then we don't know what kind of phage could use for that specific new bacterium that potentially, pathogen or potentially we want to isolate. So given all kind of challenge I just mentioned, I just wanted to know what would be your vision to really enable this kind of holy grail type of experiment in the future. That's one thing I wanted to hear from you to conclude today's seminar.
Yeah, I mean, it's a really important question. I think the holy grail is maybe a little hard to describe. It's effectively asking for a silver bullet, which I think I view as more of a platform technology that involves many approaches simultaneously. And so I do have a slide that I can just quickly put up so people can see it. But it basically addresses all of several key compartments of what we think are barriers to efficient microbiome editing, really in any species. The first is overcoming physical barriers. Some bacteria protect themselves in biofilms with capsule polysaccharides, for example. Mobile genetic elements like phages, conjugative systems have evolved to recognize those specifically and use them as an advantage for specific injection of their DNA. And so I think approaches will probably largely need to be tailored towards specific bacteria in this way. And then once DNA enters the bacterial cell, it's immediately challenged for restriction by things like restriction enzymes, CRISPR-Cas enzymes. And if you can inhibit these types of responses, you can increase your transformation efficiencies by 5 orders of magnitude. And so that's very important. That's something we're also thinking a lot about and strategies that can overcome that. We know that oftentimes our tools, if we want them to work, we need to get them on a stable vector that can replicate and maintain itself long enough to express and find a target site and edit. And so we have an approach where, we're basically mining plasmid diversity with colleagues here at Biome, Carlotta Ronda, Spencer Diamond, Ben Rubin, and others, where we can apply these in library format and access many things at once and see which plasmids replicate in which bacteria. We're constantly trying new editors. We know some editors work in some cells and not others. That might be directed evolution, it might be rational engineering, or might just be trying other natural systems. And then finally, let's say you can take that one weird bacterium you were describing, Tae-Suk, and get one edit in the context of a microbiome. Say it's not isolatable, but you can edit it in the microbiome context. How do you get that bacterium that's been edited to kind of enrich and take over the unedited fraction of that population? You can't use antibiotic selection markers. And so we're thinking a lot about enrichment schemes that can use a genetic handle to facilitate that sort of an approach. And so this is what we're thinking about. And it's not, unfortunately, it's not a holy grail, but we think we're going to learn a lot about each of these compartments and at least figure out what the best strategies are for each of them.
Fantastic. Yeah, fantastic. So again, I'm sorry for my technical difficulty. I don't want to turn off my computer. It may kind of completely all the people get off, from the, so I couldn't join, or somehow I couldn't turn on my camera and couldn't turn on my microphone, but somehow magically that solved. So I was so sorry about that, but brilliant talk, amazing talk. I really appreciate your wonderful presentation. Lindsay, do you have any other thing?
No.
Yeah, because we spent already 20 minutes after 11 Central Time, or now my time is 1:20 a.m. So we need to close. Let's end. So thank you all for joining and staying today. We'll meet again on October 17, Thursday, the same time, the same Zoom link. We'll have Professor Bob Langer at MIT and Dr. Layli Lohani also at MIT. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. Thanks. I stop recording. Just 1 second.